Hello. All right, so welcome to today's meeting. We're going to talk about a number of things. I wanted to highlight some media on YouTube and things like that. And then I wanted to talk about a paper on C. elegans that just came out. So the first thing is congratulations to Varsith. And Varsith made a pull request to the DevoLearn repository. So this is number 40, add hypersage and unisage comms, improve config and edge handling. So this was merged by Lalith. And so congratulations to Varshith for making that contribution. He puts in the comments here, introduces hypersage conv and unisage conv. These are convolutional layers for hypergraph convolution. Updates config to auto-generate embedding paths, and support new conv slash recurrent neural network types. And improves hyperedge construction by using pre-computed birth times for live cell filtering. Updates argument parsing and embedding saving logic to reflect new config behavior. Refactors model to support new convolution and RNN cell types, and updates snapshot construction to past birth times. So this is on the work that I guess that Lalith had done over the summer, and this is just issuing a pull request to make some of these updates to those tools. And so Lalith says, overall good work, needs some minor adjustments. Once they're cleared, we're already merged to the main. So that's what's going on there. Again, this is in the DevoLearn organization. This is the DevoGraph repository. I also wanted to point out that ALife 2025 is now on YouTube. So ALife 2025 conference, which was this fall, and this is the website. They put up their videos. So they recorded their videos. Now they're ready to view. So there are a number of sessions. There's evolution 1, 2, and 3. Here are the keynotes. So, Hector Zenil was one of the keynotes. I think Michael Levin was one of the other keynotes. And then, of course, they had a robotics session. And they had these panels where they had different conversations. This is the Antikythera conversation. Antikythera is an ancient Greek computer that people talk about a lot as being one of the first computers that humans created. Of course, those are all ancient computers, and they did things like calculate numbers and manipulate symbols. And of course, this is far removed from the computers we have today, but it's still worth thinking about in the context of modern computing, especially fields like ALife, where you're trying to understand the origins and evolution of life. So check that out on YouTube. ALife 2025 is the channel. All right, now this is the paper here I want to talk about. This is from PNAS. This is a number of different authors on it. This is Victor Ambrose, Marty Chelfie, Gary Ruvkun, Nathan Schroeder, Paul Sternberg. These are, and what they're trying to do here is they're trying to understand, or they're trying to give us this path from the origins of C. elegans as a model organism to the Nobel Prize that was awarded last year on to Ambrose and Chelfie for their work in C. elegans. So C. elegans originated as a model organism in the 1960s. Sidney Brenner proposed that we could use C. elegans to sort of model a lot of things in biology that we hadn't been able to model previously. There are a number of properties of C. elegans that enabled all these types of investigations. Everything from highly tractable genetics to a very small connectome and a very short generation time. So we can do a lot of things with C. elegans that we couldn't, say, do with humans or even other model organisms like mouse or even fruit flies. And so C. elegans became a predominant model organism. It went from like the lab of Sidney Brenner, and then some people at Oxford who started working on it and became famous, to a model organism where there's a community of several thousand people, and the Worm Conference, of course, is the manifestation of that work, where you have it's a very large conference, where you have all of the people using, working with C. elegans in very different contexts. Some people are geneticists, some people are interested in the connectome, some people are cell biologists, some people are evolutionary biologists, and so forth. Some people are medical scientists, and they're all interested in using C. elegans because it's easy to culture, because it has all these different life history and physiological traits that make it attractive to being a model organism. And so this is basically an overview of that history, and then, of course, a lot of the community tools that have enabled the growth of C. elegans as a model organism. So one of the things you find in the C. elegans community are these great curated resources. Things where you have data repositories, and you have libraries of mutations, defined mutations. You have libraries of strains of worm that you can order and bring into your lab and start culturing them immediately. You have knowledge bases where you can find out information about any aspect of C. elegans biology that you'd like. Worm Book is one example of that. So you have all these different, and then you have atlases where you can look up parts of the anatomy, and you can look up how they register gene expression and all of that. So there's a lot of interesting stuff that you can do with C. elegans, a lot of useful things. It's a very well organized community, and so they kind of walk through a lot of this work, and of course, a lot of it's supported by the NIH, and so this is tied in very tightly with medical research. So a lot of disorders that we have in humans, we can actually look at the homologs and paralogs of genes in C. elegans, and we can understand the genetic bases of these diseases in C. elegans. We can do experiments in C. elegans we couldn't do in humans, and we can figure out maybe some of the signaling pathways involved in these disorders. So it's a very useful tool. So let's go through the abstract. So the abstract reads, experimental organisms such as the nematode C. elegans or Caenorhabditis elegans are fundamental to biological discovery. So we have all sorts of model organisms out there. Sometimes we have model organisms for specific modes of biology, like developmental biology. So we have zebrafish, and we use zebrafish for developmental biology. We use other types of organisms for developmental biology as well, like Xenopus. We also use C. elegans for developmental biology, but not exclusively. We use fruit flies for looking at genetics. We also use C. elegans. And so if we go across our model organisms, sometimes they're very specialized in terms of what they're used for. Sometimes they serve as a sort of workhorses. So like C. elegans is similar to mice or to yeast in that it's this workhorse where it's just doing all sorts of, you can do all sorts of experiments with it and really get down to pathways, molecular pathways that can give you information about a wide host of biological processes. So the success of C. elegans research has been greatly enabled by infrastructure that allows thousands of scientists to share and access research materials and unpublished information efficiently. Here we celebrate the worm by interweaving vignettes describing 4 Nobel Prize-winning discoveries, with descriptions of how the major NIH research resources and those 4 are actually, and those 3 are the Caenorhabditis Genetics Center, Wormbase, and Worm Atlas, provide invaluable support for all C. elegans research. The synergy between investigation, and the availability of shared resources for the C. elegans community is a paradigm for all model organism research, and the continued support of such community research resources will be essential for maximizing impactful discoveries in the future. Very good. So, really, when we think about our knowledge of human biology, we think of studying humans. We think of medical imaging. We think of things like cadaver studies. We think of things like direct assays. And that's all useful, but sometimes we need to understand processes that we can't directly observe, or it's very hard to directly observe. And so this is where, of course, our animal models come into play. And they're very important parts of the biomedical enterprise. I know people are trying to sort of get around that with cell models, but I doubt that cell models are really going to give us the kinds of insights that animal models will. And so animal models provide unparalleled experimental access to fundamental biological processes that are shared across evolution, including with humans. The logic is simple but profound. If a mechanism is ancient and conserved, then uncovering it in other organisms provides insights into our own biology. So this is an important point, is that we can use the power of common ancestry to understand a lot of these processes at a very fundamental level. So, of course, humans and nematodes they share a common ancestor. And that common ancestor had genetic pathways that diversified in both lineages, but have commonalities in terms of their structure, in terms of signaling that is insightful, that can tell us something about. Like if we knock down some gene in C. elegans, we can see an experimental outcome. We can try, and we have the tools now to find corresponding pathways and genes in humans, and then do that sort of maybe we can create drugs or some sort of intervention to help in human health. And so it provides great insights. And of course, in aging, that's a famous system in C. elegans, metabolism, and other types of processes that are very general are, those are very, we've had some pretty impressive results from that. This has proven true time and again. Discoveries in yeast, flies, worms, and mice have illuminated the foundations of human development, physiology, and disease. The best established models share certain features, including the ease of cultivation, a rapid life cycle, relative simplicity, and accessibility to genetic and genomic analysis and manipulation. These features facilitate both basic research and exploration of potential translational applications in a controlled and cost-effective way, free of many of the ethical concerns that apply to any research using humans and human tissues. But these organisms are not simply substitutes or models used to study known human biology. Often, as outlined below, they are the source of new discoveries that illuminate unforeseen aspects of human biology. So this is just kind of getting into how there's this, this rationale for using C. elegans as a model organism. But of course, just having a model organism is not enough. Just because you can culture it and you can do experiments in it, that's fine, but you also need a support structure in that community that emerges to really make a model organism effective. And so in this section, they go through a brief history of C. elegans as a model organism and the worm community, in quotes, because the worm community is very important to have people who can share data, who can share insights, to have a nice conduit into the community. So when you start working with C. elegans, you have those resources that help you along. There are a lot of people who maybe start working with a model organism and they'll try culturing it. I think this is true of a lot of diatom people, where they start working with diatoms and they're able to isolate it and grow it in culture. But then it's very tricky to maintain the cultures there's all sorts of knowledge that isn't like immediately available, and it's very frustrating to get your some actual quality work established in a setting like that. So in the C. elegans community, you benefit from a lot of communal knowledge and having that at your fingertips. So the use of C. elegans to dissect biological truths originated in the visionary thinking of Sidney Brenner. After an extraordinary career defining fundamental principles of molecular biology, by the early 1960s, Brenner was searching for a relatively simple animal where these principles could be applied to understand mechanisms controlling development and nervous system function. He chose C. elegans for its simpler anatomy, development, and more rapid life cycle than the well-established model organism Drosophila melanogaster. So Drosophila, the fruit fly, was a model organism from well before when C. elegans was established. A lot of the early genetics experiments were done in fruit fly, and there are all sorts of uh, advantages of using Drosophila as a model, but of course it's more complex. The development is longer, and the life cycle is longer than C. elegans. So there's an advantage to choosing C. elegans over Drosophila. Of course, Drosophila had well-characterized genome for the time, so that was another advantage. So C. elegans had a lot of innate advantages, but it had to overcome some barriers to really kind of getting out quality experimental. While considered by some to be an odd career move at the time, Brenner's decision propelled a little-known roundworm into a juggernaut of biomedical research. And then there's a figure 1, we'll see that in a minute. Since Brenner's seminal publication establishing the worm as a genetic system, research into C. elegans has grown continuously and now includes fundamental discovery in myriad areas, including landmark discoveries recognized by 4 Nobel Prizes. So there have been 4 Nobel Prizes given for different topics in medicine and physiology that have involved C. elegans in some way. So it's become very indispensable for discovering things, unlocking knowledge, and so forth. Brenner's use of the C. elegans attracted many researchers, several of whom established projects that produced an unprecedented look at an entire multicellular organism, mapping the entire lineage from a single cell to the adult stage, describing the cellular wiring diagram, which, this is the connectome, all visible chemical and gap junctions for the entire 302 neuron nervous system, and developing a genetic and physical map of the genome as a prelude to generating the first complete DNA sequence of an animal. And this is, of course, the C. elegans genome. So for those references that we just talked about, 3, 4, and 5. 3, of course, is the embryonic cell lineage of the nematode C. elegans. This, of course, was John Sulston's work with John White, and this was published in Developmental Biology. Then we had, of course, John White and Sidney Brenner on this paper, The Structure of the Nervous System of the Nematode C. elegans. So the first one, reference 3, talked about the lineage tree in development. Number 4 talked about the 302 neuron connectome. And then number 5 is from the C. elegans Sequencing Consortium. And of course, that's a genome sequence of the nematode C. elegans, a platform for investigating biology, and that was published in Science in the year 1998. So a few years before the draft human genome came out, we had a draft sequence of C. elegans. And of course, that's been useful in understanding the defined mutations that we have available to us in C. elegans and mapping that to a genome and then finding paralogs and homologs in the human genome. And of course, the worm's genome is compact. It doesn't have as many chromosomes as the human genome, but it encodes basically the same type of regulatory machinery that you find in humans. So it's as complex as the human genome in terms of regulation. So it has a comparable level of complexity in terms of regulatory mechanisms that the human genome has, but it's far smaller. and found on far fewer chromosomes. The development of common resources and the belief that research findings in mutant strains should be freely shared has propelled worm research to the forefront. This is a great part about like the C. elegans community, but also you see this in the Drosophila community as well, where you have this access to different mutant strains. So, you know, there's been a lot of work on creating defined mutants, which involves a lot of work on isolating the expression of certain mutant alleles and then back crossing those lines to make sure that there's total penetrance across the entire line in terms of that allele, and then being able to raise mutants that all share that mutation. And oftentimes those mutations have functional consequences. So a lot of times, you have mutations that are deficits in metabolism or sometimes enhanced metabolism. You have deficits in egg laying. You have deficits in other types of areas. You have accelerated aging. You have retarded aging. And then, of course, you have mutations that enable sort of rearing at different temperatures. So you have all sorts of different functional consequences of these defined mutants, or these defined mutations. But the great thing about it is that you can share those lines with other researchers. So there are these banks where they freeze down certain strains of worm, and then when someone wants to do an experiment with a defined mutant, they can just simply go to that bank, take out a strain, and grow it grow it according to protocols that are also shared, and it provides you with a very easy experimental testbed. You don't have to do all that work yourself, and it speeds up science. This community spirit was alive early on with Bob Bedker's establishment in 1975 of the Worm Breeders Gazette, a newsletter for sharing information prior to publication. So this is a nice resource where you can get all sorts of informal information about how to do cell culture and how to maintain your lines and your strains in a way that's standardized across labs. And it's very cell culture is very hard to do correctly, or in this case, worm culture. There are a lot of things that are sort of qualitative, a lot of tricks that you have to follow, and sometimes you follow everything to the letter and they still fail or they still get contaminated. So it's very useful to have a resource like this where you can kind of test things out and exchange sort of anecdotes and see what works and see what doesn't. So this is figure 1. Here's an adult worm and next to 2 eggs. So these are the eggs. These are embryos are contained in these eggs. And these eggs will hatch at about the 600 cell stage. And they'll continue to grow into different stage larvae, which then continue to grow into these 959 cell adults, at least for the hermaphrodite. So you have a hermaphrodite worm here, you have the eggs. These eggs will hatch, and the little larva will uncoil, and it'll grow and go through these different larval stages and then become an adult, which then can lay its own eggs. Those eggs then will give rise to several hundred worms, and then the worm will die and you'll have another generation. And that whole generation takes only a couple of days. You don't need to wait that long to get a whole generation of worms. And so it's very amenable to things like experimental evolution, where you can have defined mutants, or you can put the worms in different environments and see which ones survive and which ones don't. So there are a lot of things you can do here in C. elegans. And you can do, of course, experimental evolution, famously in bacteria, but also in yeast. But C. elegans offers this trade-off where you have this very short generation time with this relatively complex physiology and anatomy. And in that sense, we can look at things that are familiar to humans. So we can look at things like metabolism, or we can look at things like neurons, or we can look at things like a connectome. and they have some semblance to humans. And so then this is a picture of Sidney Brenner, who started doing this enterprise. And then this is the growth in publications of C. elegans work. So if it says C. elegans in the title or in the, I guess in the abstract, it gets indexed. And so you can see, though, very clearly that the number of publications has basically grown. Well, it grew exponentially in the 80s and 90s and 2000s, and it's kind of reached a plateau in the 2010s and 2020s. But that's 120 or 12,000 publications a year. That's not bad. So this is definitely a robust area that people have really found a lot of value in. And so, one of the traditions, of course, in the field is sort of sharing clones, but also in data sharing. And so, uh, John Sulston, in his work on the lineage tree in C. elegans, was a famous example of where those data were shared and where there was a lot of advances made, not just by Sulston's group and Sulston being originally in the Brenner group and then moving on to his own work, but many other groups around the world. So a lot of these early people who originated in the Sidney Brenner group moved on to graduate students, and then they, shared their data and their resources with other scientists, and they established this tradition in this community, and it allowed us to get uh, C. elegans as being the first animal with a sequenced genome, as well as leading to this tradition of open data sharing that influenced things like the Human Genome Project and other large community projects that have moved human biology forward. So the Human Genome Project started in 1990. A lot of the genetics work in C. elegans predates that. So a lot of the traditions that happened in the C. elegans community were seen as important to any sort of integrative biology enterprise. And so the Human Genome Project incorporated a lot of these traditions of data sharing and other types of community-based data sharing and information sharing. And then, of course, as we moved on from the Human Genome Project, there have been other projects like the ENCODE project where you've had the same kind of focus on informatics and on data sharing and having this open community. So they highlight 3 community resources here. This has been critical for the success of C. elegans research. And the first is the Caenorhabditis Genetics Center, which curates and distributes strains. The next is Wormbase, which is an online compendium of C. elegans molecular knowledge. And then Worm Atlas, which is the online resource for cellular and anatomical data. These resources have connected independent labs to push science forward synergistically, making fundamental and unexpected advances that would otherwise have taken decades, providing a great return on investment. So they're going to start talking about the Nobel Prizes now. So the first Nobel Prize was in 2002. And this was for the discoveries concerning genetic regulation of organ development and programmed cell death. So this Nobel Prize was awarded to Sidney Brenner, Robert Horvitz, and John Sulston. And this was, of course, back in 2002, where they basically awarded the prize to C. elegans people who demonstrated aspects of development and aspects of apoptosis. So, of course, Sulston was sort of the person who first established C. elegans as a model organism. He articulated a clear and prophetic vision: identify every cell in the worm and trace lineages, investigate the constancy of development, and study the genetic control by looking for mutants. And so that guided the work. And of course, this had an antecedent in microbial genetics, where we have these very tight and advanced analytic methods that you can't necessarily do, say, in humans, or at least at the time you couldn't do in humans, or in other model organisms like mouse, or even in Drosophila to some extent. So this is really what we were trying to do here. Taking advantage of that transparency, microscopic size, and small cell number, Solston heroically and meticulously traced the pattern of non-gonadal cell divisions. This is, of course, the lineage tree, from the single fertilized egg to the mature adult, which, in conjunction with the somatic gonadal lineage, define the first and to date only complete cell lineage of an animal. Solston described a largely invariant cell lineage that produced, for the hermaphrodite, 1,090 somatic cells, but remarkably only 959 of these cells were present in the adult. The other 131 initially looked like typical cells, but quickly underwent a series of stereotypical morphological changes that culminated in their disappearance. So the lineage tree produces more than 959 cells from the point of the one cell fertilized egg to the adult. And what happens, of course, is we have 959 in the adult, and we always have 959 in the adult. C. elegans is what they call a eutelic organism, which means that there's an invariant number of cells, unless you're talking about a defined mutant, an invariant number of cells in the adult hermaphrodite. So the cell lineage tree overproduces cells for the adult. So those 131 cells, the difference between 959 and the total output of the lineage tree, are cells that undergo programmed death. They basically die a death that is planned, it's programmed, and then maybe the question is why? Well, there are a lot of functions for cells that cells live and serve their purpose and then die, and that's exactly what's happening here. Programmed cell death, also called apoptosis, had been known to be a widespread aspect of animal development. So if you look at a tadpole's tail, they often overproduce cells and then regress back towards a final shape, or the formation of fingers and toes, where your hand is this web when you're very, very early embryonic development, and then the cells in between the digits will undergo programmed cell death when exposed to a transcription factor that basically defines the shape of the fingers. So your ancestors, phylogenetic ancestors, had webbed hands, basically, or webbed front limbs. And so you lose those cells to get the digits that you have on your hand. And this involves apoptosis. Those cells die off in a programmed way that they don't in, say, your amphibious cousins. So this is all kind of where we see this in nature. And of course, we see this throughout nature. But of course, we want to understand this from the standpoint of a lineage tree, of a developmental lineage tree, and what happens to those cells. The challenge was to determine the mechanistic basis of this striking phenomena. The starting point, as for many important studies using model organisms, was a chemical mutagenesis screen for mutants that disrupt a specific biological process. Solston identified the first cell death gene, NOC1, which encodes a nuclease that degrades the DNA of the dying cell. A few years later, Edward Hedgecock identified mutations in two genes, SED1 and SED2, recorded for the phagocytic engulfment of the dying cells. In mutants that lack SED1 and SED2 function, the cell corpses persist. So this is all kind of involved a huge genetic screen, and they were able to find out what the mechanism for apoptosis was, and that this is conserved across evolution. So this leads us to understanding maybe how cell lineage abnormalities play a role in organ development and the genetic regulation of organ development, but also leading to novel insights into different signaling pathways such as EGFR, RAS, ERK, and Lin12 notch pathways. Focusing this approach on apoptosis, Ellis and Horvitz performed a screen for mutations that eliminate the persistent corpses of SED1 mutants, reasoning that such mutants might include those in which the cells abnormally die and instead live. This screen and related screens were spectacularly successful, identifying the core molecular genetic components of an apoptotic pathway that is conserved across the animal kingdom. 3 pro-apoptotic genes, EGL1, SED4, and SED3, promote programmed cell death, while a single anti-apoptotic gene, SED9, prevents programmed cell death. So this is kind of going over. This is a graphic that shows the conserved pathway for programmed cell death, where you get this core pathway that's activated for the kill. It involves these different genes here, EGL1, SED9, SED4, and SED3. SED-A activates 2 other pathways, which is where we have this pathway that engulfs and removes the dead corpse. So it has this phagic action that involves PSR1, SED5, SED2, and SED12. And then they found the human orthologs. So these orthologs are found in humans, but they were discovered in C. elegans screens first, which are SED1, SED6, SED7, and SED10. And so those all work together to remove the cell to make sure that doesn't, kind of stick around. We don't like to have dead bodies around in our community. Same holds true for cells. And so that's what this is. So then they highlight the Caenorhabditis Genetics Center that was established in 1979. This is a place where you can go. It's a stock center where you can order different lines of C. elegans to find mutants. The Caenorhabditis Stock Center not only accommodates strain requests, but also involves access to research and education and increasing that in the area of not only C. elegans, but in genetics as well. So over 4,100 individual labs have obtained strains from the CGC within the last 10 years. More than 1,600 of these have official worm lab designations required for naming strains and wheels. Many of the other labs are primarily focused on other organisms, but look to the worm to explore homologs of their favorite genes. Often they can find strains pre-made and ready to order, streamlining the efforts required to test a hypothesis. So the next Nobel Prize was in 2006. This was for the discovery of RNA interference, gene silencing by double-stranded RNA. So this is using RNAi. This was awarded to Andrew Fire and Craig Mello. And they led research groups that identified double-stranded RNA as a potent and specific trigger for RNAi and consequent gene silencing in C. elegans. So RNAi silences genes, and there's this trigger, which is double-stranded RNA, that allows for this process to happen. So this discovery uncovered an ancient guided search mechanism that cells use to recognize and regulate genetic information. These types of RNAi and other types of small RNA pathways are universal. So this isn't just C. elegans specific, but happens across, the tree of life, governing gene expression, protecting genomes, and shaping heredity. RNAi has become a valuable tool for high-throughput functional screens and individual intervention experiments. While this role has been shared with genome editing methods such as CRISPR in more recent years, both workflows share the features of combining production of limited or massive libraries of RNA guide sequences that target specific genes with methods for introduction into cells, phenotyping, and assessment of causal relationships. So RNAi can be used to silence genes. We can use CRISPR and other methods to more in a controlled manner slice out genes or cut out specific stretches of DNA. And, but still you have a lot of value in RNAi-based screens, which are standard for functional genomic approaches in cell biology, genetics, and biotechnology. And of course, it did serve as a paradigm for the CRISPR methods that we have today. And so really the RNAi award was built on this whole idea of how, people discovered how double-stranded RNA structures participated or set up this gene silencing process that people didn't really understand before. Being able to understand the structural requirements for RNA-based silencing occurred through the use of the C. elegans model system. Then they talk about worm-based, which was established in the year 2000, and the Alliance of Genome Resources established in 2016. So again, C. elegans was the first animal for which a whole genome sequence was available. So we had, before the C. elegans genome, we had bacterial genomes. Those bacterial genomes were circular and rather small in size. The C. elegans genome was the first metazoan genome. We would get the human genome a couple years later. We'd get yeast genomes, we'd get mouse genomes, we'd get all sorts of genomes in animals. But C. elegans was the first whole genome that was created for animals or sequenced for animals. And so this is where, we had this happen in a model organism first, and then we had our human, and we had to have the model organism genomes. And in fact, that was a major part of the Human Genome Project, was to create these analogs in model organisms so we could use some of that information to inform the function of human genes. Just having the sequence isn't enough. We need to understand the function. And in C. elegans, that's fortunate because we can do experiments where we can target specific genes and specific pathways, much easier than we can do in humans. So we find, though, that nearly 40% of C. elegans genes have human orthologs based on extensive primary sequence conservation. So we have a lot of shared genetic material, shared genetic function between C. elegans and humans, and we can use that in, looking at human health or looking at physiology or some sort of pathway that might tell us something about, say, like apoptosis in humans. In addition, many additional worm genes are functional orthologs of human genes, recognizable by conserved domain structures and or AI-assisted structural predictions, exemplified by Lin14, a direct target of the Win4 microRNA. And this, of course, is part of the 2024 Nobel Prize, for which two of the authors won. The genome sequence is generally a starting point for the creation and study of human disease models and is integral to all studies of aging, neurobiology, development, and physiology with applications to human biology. And so the third Nobel Prize was in 2008. And this is for the discovery and development of green fluorescent protein, or GFP. So GFP is actually something that occurs in nature. It occurs in a marine organism, and it's generated. There's GFP, and then there are other types of fluorescent proteins like RFP, which is red fluorescent protein, YFP, which is yellow fluorescent protein. And these proteins have been isolated from the natural populations and brought to the lab to use as biological markers. So the idea is you will take like a cell or organism and you'll infect the cell or the organism with this fluorescent protein. You'll have a construct, you'll infect the cell, you'll deliver the fluorescent protein into that cell, and then it will bind with some, you'll have a binding sequence that's associated with the fluorescent protein, and it will bind to some, say, RNA of interest or protein of interest. And then once it binds to that thing, it will show up in, say, like if you're doing some sort of fluorescent imaging. So you have fluorescent imaging. It's usually in the ultraviolet color spectrum, and you can actually see these markers as brightness. You see them as green brightness or red brightness. So it makes it very easy to localize a lot of things that are expressed in cells. So this is, of course, an example of how GFP is used in C. elegans and in human cells and in organoids. So this is touch receptors across human cells. You get, things that identify a touch receptor, and the green fluorescent protein is expressed in those receptors, and you can see them here along the lateral lines of the worm. So this shows this map, and we can actually localize these cells by that signal. And you can take these images and you can co-register them with some sort of anatomical atlas, and you can have spatial specificity at a pretty good resolution with not a lot of noise. There's things like autofluorescence and other things that can be corrected out using imaging software. But other than that, it's actually a pretty indispensable tool for localizing different cells and different RNAs and different proteins. This is the worm brain, where we have these different fluorescent proteins that are used to localize different cell types. You can see the different connectome cells here in the anterior part of the worm. And again, we're identifying this by the fluorescence level. So, the fluorescence level is often an important indicator as well. If it's weakly fluorescent versus highly fluorescent, and we can measure and quantify that fluorescence as a measure of, say, how abundant a certain RNA is or a certain protein is. And that tells us something about what's going on in the cell. Is the cell producing a lot of the substance or very little? And maybe that's a sign of like strong affinity or weak affinity for a certain cell type. There are all sorts of ways that we can do this. And you see this in papers that we talk about in these meetings where you have where people try to quantify the signal and make statements about their sample and or about their treatment condition based on the fluorescence indicators in that image. So then this is actually where we have green fluorescent protein with cherry red stain, and it defines different aspects of human stem cells, stem cell aggregates, and human organoids. So we can use green fluorescent proteins in combination with different stains. And so this is kind of how this will look under a microscope. You can combine signals and it can give you different pieces of information localized to different parts of a cell or different cells or different parts of what's becoming a tissue or an organ. So then they highlight Worm Atlas. And Worm Atlas, of course, is an anatomical atlas. And there was a sort of a goal of creating something that's similar to an atlas of human anatomy, but for C. elegans. And this is an example of the atlas where you take images like this and you create these three-dimensional images like this. So you're taking something that's very hard to see and understand in a microscopy image. I mean, you can see the structure, but it's hard to like visualize what that should look like in anatomical context. You see a very faint worm body here, but then this nerve coming up the interior to the anterior end of the worm from this neuron AWCL. So, that's kind of hard to understand and interpret. And so we can turn that into an illustration. Here we have, not only the nerve, but the sheath of the nerve, and then the cell, and then the terminus at the edge of at the anterior edge of the worm, and then the worm body around it. So we can understand all this in context using an atlas. We can also play with the atlas in ways we can't play with the image. And we can also annotate the atlas in ways that we can't annotate an image. So we can combine different cells, combine different nerves, and annotate them, differentiate them, explore them differentially, and so forth. And so this is other examples of the atlas where they have the slidable worm where you go through different slices of the worm at different lengths of the body and see what's there. You can also look at the atlas in terms of labeling things so you can understand where cells are in context, where cells are in the body of the worm. So this is a very useful tool to have because you can actually go there and think about your problem. Think about, if you're looking at a specific cell or a specific anatomical location and how that sort of translates more generally to the rest of the body. And so, the thing about C. elegans is that's not the only nematode. There are other nematodes. There are other nematodes that are model organisms. There are other natural populations of nematode. So there's the satellite model nematode Prestionchus pacificus and a human parasitic nematode Strongyphites styrocoralis. And so these are different nematodes that have different numbers of cells in the adult. They have different lineage trees and all of this. So there are differences in terms of different nematodes that are actually pretty closely related. And so, this is something where people are starting to look at these other nematodes as model organisms for different things. And they want to make comparisons with C. elegans. And so it's important to have these resources not only for looking at human disease, but looking at these closely related nematodes and looking at the differences there. Finally, we have the Nobel Prize in 2024. This is last year's Nobel Prize for the discovery of microRNA and its role in post-transcriptional gene regulation. So this is, of course, where we have our microRNAs, not RNAi. It's a different type of RNA. It's not mRNA, it's microRNA. So microRNAs are very short RNAs that play a role in this post-transcriptional gene regulation, where you have translation, of course, which is where you turn a DNA sequence into an RNA sequence. The RNAs will form secondary structure. Then they go to be transcribed at the ribosome. And then the ribosome, they get turned into amino acids. And then there's this gene regulation before the assembly of the proteins that happens that modifies those transcripts even further so that the protein looks very different from the gene sequence that produces it, or the gene sequences that produce it. So microRNAs are small non-coding RNAs that play an important role in post-transcriptional regulation of gene expression in plants and animals. So microRNAs are widely applicable in life, in plants and animals, both. Not in bacteria as much, but this is just a, this is still an important discovery. The discovery of microRNAs emerged as an unexpected outcome of the Ambrose and Rivkin labs studies during the 80s and 90s of the Lin4 and Let7 genes of C. elegans and their roles in controlling the timing of larval developmental events. So this is where we, of course, have these larval stages we have going from L1 to L4, and there are these different, courses of development that can occur. You have in an organism where the adult always has the same number of cells, at least in the hermaphrodite, you have a lot of changes or adaptations that can occur. So you have a starvation that you can have after L1. You have the Dower adaptation or the Dower phase, which can go from L2 to L4. And these are all responses to extreme environmental conditions. Furthermore, during each larval stage, you have cell growth, you have morphogenesis, and you're adding cells at that time. So there are a lot of things going on in the larval stages that are quite interesting in finishing off the adult worm from what's hatched. And so a lot of these things involve post-transcriptional regulation. And so these involve Lin4 and Let7. For a half a dozen years after publication of the 1993 paper reporting the discovery of the first microRNA, Lin4, it was not clear whether this little non-coding regulatory RNA was merely a specialty of C. elegans or representative of a broadly conserved class of RNA regulators. And of course, it was the latter, or else they wouldn't be getting a Nobel Prize. The 1990 discovery that a second C. elegans microRNA, Let7, is conserved in sequence across all bilaterian animals, putting all doubts to rest, and in short order, hundreds of miRNAs, not mRNAs, but miRNAs, were discovered across the eukaryotes, including humans. Strikingly, Let7 is not only the only microRNA to be deeply conserved across animals, and in some cases, even the specific target genes of conserved microRNAs are conserved over vast evolutionary distances, emphasizing the fundamental importance of microRNA-mediated gene regulatory mechanisms. So why did we find microRNAs in C. elegans first? Well, we can work, of course, we're working with worm mutants, and we're able to do these kind of unique mutations. So Lin4 and Let7, as we have multiple defined mutants of each, they exhibited developmental timing phenotypes. So these are temporally disjointed expressions of juvenile and adult somatic features in the same animal that, of course, because you have a very short generation time, you can observe those under the microscope and pretty quickly put that together. And so that's how they were able to discover this. This is an example of conserved microRNA target regulatory motifs. So you have these three processes on the left. You have what happens in the worm in the middle in red, and then what happens in humans on the right in blue. So for morphogenesis is a process, in the worm you get microRNA 51 and microRNA 100, and that puts a break on HST 3.2. And so in humans, we see a similar thing with microRNA 100, putting a break on HS3ST2. So this is not exactly, it's homologs and paralogs acting the way they do. Then muscle physiology, which is another process, uses microRNA1, and it puts a break on these VHA genes, VHA1, 5, 11, 12, and 13. And then in humans, we have MIR1 acting to put a break on ATP genes, ATP6V, and different variants of that. So again, we have different named genes, but they're related, and the pathway is conserved because it's involving MIR1 in both cases, and it's being sort of, it's putting a break on these other the expression of these genes. So it's actively suppressing these genes post-translational. Then developmental timing, we have in the worm, we have Lin4 MIR125, we have LET7, that puts a break on Lin28. And when you put a break on Lin28, that puts a break on the L3 fate and extends the L2 fate. So the L2 fate is the second fate, larval phase. It's extended when Lin28 is suppressed. L3 occurs later, and you get this extension of the L2 fate. This is important in the Dower phenotype that you see in development. Then in humans, we have MIR125 and LET7, so the same sort of pathway. We have Lin28B in humans, and that puts a break on late competence and extends early competence. So you're extending an earlier developmental fate in the same way you're doing this in the larval stages. So it's fascinating that we have this sort of conservation, even though the life history is very different. And, we don't have larval stages in humans, but we have this sort of, we still have development, and it still comes from the same places. But these microRNA regulatory mechanisms seem to be pretty consistent across a wide range of the tree of life. So they conclude by saying that the past half-century of C. elegans research compellingly demonstrates how fundamental investigations using an experimental model organism can reveal conserved biological mechanisms and greatly impact our understanding of human biology, disease, and the ways to develop novel therapeutics. It is fair to say that all studies of C. elegans have been greatly enhanced and indeed would not be possible without the NIH-funded shared resources of the CGC, Worm-Based Alliance, and Worm Atlas. And if history is any guide, the next half century will bring insights we cannot even imagine today, insights that will again transform our understanding of biology and our ability to treat disease. So this is great. This is a nice sort of, I think, piece of public communication as much as a review paper in telling people how important C. elegans research is, how central the NIH has been to this enterprise, and then how wide-ranging, you work with a nematode. You say, that doesn't seem to have any consequence. And of course, it has great consequence, but you have to let people know what that consequence is. They have to understand what the impacts are. And indeed, there are a lot of impacts here. So that's all for today. Thank you for watching to this point, and I hope you learned something.